Welcome everybody to the Michigan Molecular Modeling meeting. Today we're thrilled to have Xiaorong Liu present. Xiaorong is a postdoc in the Brooks lab and will tell us about high-throughput free energy methods in drug discovery. So this will be about a 20-25 minute talk, and hopefully we'll have some good questions. And then after that, we'll have a chance to talk about perhaps a hackathon that we might run this summer. So please take it away, Xiaorong.
Thank you very much, Matt, for your introduction. I'm thrilled to present my work in this meeting. I have enjoyed it very, very much, and I'm very happy to present my work here. So today I'm going to share some method development and application of high-throughput free energy method in drug discovery. Let me see. Okay. So I guess here I probably don't even need to emphasize how important computational methods are in drug discovery. So for example, in this high throughput stage, usually there are a lot of methods are very efficient. For example, machine learning and docking, they're highly efficient in terms of searching the large chemical space. Okay. But if we want to go to need optimization stage, usually it requires a highly accurate computational method. For example, usually the reason is we usually have a same scaffold. We just put different decorations or substitutions at different places of this scaffold. So that's why in those optimization stage, usually for example, docking scoring function is probably not accurate enough. So, and we propose that we probably want to use this molecular modeling, or more specifically, physics-based free energy method to do this calculations. So I think you are all probably very familiar with free energy probabilities and everything, but I just probably want to take just one more slide to remind everybody about those concepts. So experimentally, we have this protein and ligand, they interact with each other and we measure the concentration of this species so as to calculate this dissociation constant. Okay, this dissociation constant, they are related to the binding free energy, of course, and it also can be computed using those probabilities there. Okay, so and this is what we are doing in computation, especially molecular modeling. More specifically, if we know the force field of the system, we can simulate the spontaneous fluctuations of the system, so that once we get enough sampling of different conformational states, we can plug into this equation so as to convert those probabilities to free energy. So I want to use a table to highlight some of those key features of those quantities. So for example, if we go to this increasingly tighter binders, of course, we have lower binding free energy and smaller KD here. Okay, the main thing is if we're talking about, for example, a tight binder in this nanomolar binding free energy range, what we are thinking is only one out of 1 billion snapshots will be in the unbound state, and all the others will be in the bound state. So this poses a great challenge in terms of absolute binding free energy calculations, because it requires us to do a lot of sampling in order to get some snapshots of those unbound states. But very luckily, what we have is in lead optimization, the quantity we care most is probably the relative binding free energy. So because this free energy is a state of function, so that's why we can construct this thermodynamic cycle. And from there, what we can see is that the summation of this quantity should be 0. And if we rearrange those equations a little bit, we can see that the relative binding free energy, which corresponds to the difference of those two horizontal lines, now can be computed using those two vertical lines. So those vertical lines correspond to the transition or transformation of ligand 1 to ligand 2 in either unbound state or bound state. Okay, so this type of transition is similar, for example, to historical concept of converting, for example, iron to gold. So that's why we call them alchemical free energy methods. So I want to review a little bit of how we do this alchemical free energy calculations. So usually what we do is we have this, we need to construct, for example, a hybrid system. Okay, this hybrid system contains some decorations, for example, the red one, and also it contains some decorations of this blue substituents here. Okay, so our potential energy is just a linear combination of those two, basically end states. And in this case, lambda, we use lambda here to control the intermediate state, and lambda will reflect the level of presence of substituents in the system. So for example, here when my lambda is 0.9, we are going to see more blue substituents here, and a little bit, just a little bit, probably 0.1, this red substitution here. Okay, so that's how we construct the system, construct this hybrid system. Okay, and we need to do a lot of these systems, or sometimes we just call them windows. Okay, and we just compute relative free energies between step 1 and step 2, and then step 2 to between step 2 to step 3, and do this, and then in the end just sum them together. Okay, so as you can see, probably from this illustration here, you can probably see that we are going to need a lot of windows in between if we want to transform something small to something big. Okay, but this has been, let me see, this type of approach has been very, very successful. For example, in Schrödinger, they have helped design this tyrosine kinase 2 inhibitors, and also another startup company called Silicon Therapeutics. They have been doing similar calculations for many years, and recently it has been bought by a bigger company for about $450 million. So they have been very, very successful. They have this type of calculations have been offering basically practical solutions to need optimization. But as you can probably imagine, one of the big challenges is the computational cost. Okay, I want to see that a little bit more. It's like here, although I only showed 5 windows, but in practice, usually we need 10 or 20 windows. And the reason is actually probably very simple. It's like we need to make sure there is sufficient overlap of those phase space so that we can get faster convergence of those calculations. And in practice, if we have charge transition or charge changing transformation, then we will probably need even more windows there. So that's the one thing related to the computational cost. Another thing is this is like a pairwise calculation. What that means is when I set up this one set of simulation, I can only do like a free energy difference between 2 molecules. So it scales linearly with the number of compounds that I'm interested in. And sometimes we may need even more, we need more basically transformations there in order to get very high precise result. So how do we basically alleviate those 2 problems? So the key idea in this what has been developed in Brooks' lab for probably more than 20 years now. So the key idea is in lambda dynamics, instead of having fixed lambda values, now we are going to treat lambda dynamically, and this lambda is going to spontaneously fluctuate between 0 and 1. And for example, when lambda is 0, it corresponds to 1 molecule. When lambda is basically in 2 end states, it will correspond to 2 different molecules. Okay. And if we collect lambda as a function of time, we can actually compute free energy by simply counting, plug into this equation, collect snapshots in those end states, plug into this equation, we are going to get the free energy there. So in this way, we eliminate basically we don't have to use so many windows in this approach. Moreover, this lambda dynamics can be extended to treat multiple substituents at multiple sites simultaneously. For example, here I have 2 sites and set 1. In set 1, I have 3 substituents. All I need to make sure is the summation of those lambdas are 1. And also at site 2, the summation of those lambdas is 1 as well. Because as I mentioned earlier, lambda sort of represents the level of presence of substituents in the system. So because of this, we can do now we are able to basically simulate multiple compounds in a single simulation, and in this way, it's very good at exploring this large combinatorial library of small molecules. So it scales excellently with the total number of compounds. And another way to think about this method is like those molecules, those substituents here, they sort of they are sort of like a mimic the experimental competition assay. The thing is here, they are just competing these interactions with their environment. So I guess, so this is the overall design of this high throughput free energy method called multisite lambda dynamics, or MSLD for short. So how does it really work? Let me see. So I will first share one story, like by asking these two questions. First, can we really use this approach to do large-scale free energy calculations? And even if we get those numbers, do those numbers really can guide us optimize a lead compound? So the system that I'm interested in here is called TSSK1B. So it's like it's an understudied kinase that is associated with human cancers, especially, for example, in this figure shown here is like if we knock out TSSK1B kinase, the cell viability decreases dramatically. So that's why there is a strong interest in terms of design inhibitors targeting this enzyme. Sorry, targeting this kinase. Okay, so my collaborator, before I start this project, actually, my collaborator already identified a lead-like compound. For example, we call it TQQ. Its IC50 is about 400 nanomolar. And also, more importantly, it actually shows inhibition effect even in cellular assays. So that's very promising. And the goal of my computational work is that can we use free energy calculations to basically further optimize this molecule so that we can get even tighter binders to TSSK1B. So what we have is that we examined these molecular structures and identified basically 5 sites to modify, and we basically change them with some substitutions there. And in the end, that corresponds to about 1,300 ligands. This is probably one of the largest free energy calculations so far. But I want to mention the computational cost. We run about 120 nanoseconds. If we use the traditional windowing approach, it probably requires about 36,000 nanoseconds. So in comparison to the traditional approach, this probably represents about 300-fold reduction in computational cost. So that's sort of very encouraging. So what do we find? Well, from those calculations, what we actually found is that, for example, in site A, if we change it to some butyl group, if we modify it to some larger hydrophobic groups, the binding free energy will be lower, the binding will become tighter. And in site E, if we add more polar groups, then the binding free energy becomes lower. We get tighter binders there. So that's what we predict from our simulations. And interestingly, this can sort of somehow supported by experimental studies as well. For example, at site E, experimentally, if we have those 1, 2, 3, those 3 substitutions here, and if we modify this from 1, make it a bit more polar, and then we can see the free energy the binding becomes tighter. This is consistent with our free energy calculations, although our free energy calculations underestimate how much the improvement should be. Another thing is because this free energy calculation is sort of physics-based, so it's very easy to interpret the result. For example, here, when we talk about site A, why do we prefer a hydrophobic group, hydrophobic substitute there? Here, what I'm showing here, this is a protein surface colored by their partial charge. So if you see a white area, that means it's a hydrophobic patch. And if you see red and blue, that means it's sort of, it has some partial charges. It's probably polar or charged. Okay. So in set one, in my set A, if I have a butyl group here, you can see that it fits nicely in this hydrophobic group. So that's why a hydrophobic like a non-hydrophobic chain is favored in set A. Oh, sorry, did I get a question? Okay. Okay, I guess I will move on. Sorry, I probably didn't hear. And then in set E, what happened is in set E, we can see we have some negative charge, for example, aspartate residues there. So that's why if we have some polar groups here, it will be more favorable. So I guess back to my, in summary, back to my the original questions that I have. The first one is, can we do this type of large scale free energy calculations? And the answer is yes. We examined about 1,300 ligands, and this is one of the large scale free energy calculations to date, probably. And also it reduces the computational cost by about 300-fold. And also, and also because this is physics-based approach, and we can easily interpret the result, and this result can this free energy calculation seems to be able to guide the optimization of each compound. We identify something that is able to improve the binding affinity, which can be supported by structural analysis or experimental measurement. So this is sort of very exciting. However, is the problem sort of completely solved? The answer is no, because one of the main challenges is we need to consider protein conformational dynamics. So for example, here I just want to illustrate that even for the same receptor, if we have different ligands bound there, the receptor needs to reorganize a little bit in order to accommodate these different types of conformation, in order to accommodate different types of ligands. So that's why considering this conformational change or conformational dynamics is very important in this type of calculations. However, there is a huge gap. I think probably you all could appreciate that it's there's a huge gap between our simulation time scale and the time scale and the biological time scale. For example, here in my simulation, I only run 120 nanoseconds, but in practice, some of those transitions can happen within a microsecond or millisecond. So that's why we need something called enhanced sampling. And the idea is, of course, we have different types of enhanced sampling technique. And with enhanced sampling, hopefully we're able to explore the free energy landscape more efficiently. Okay. So in my next slide, I'm going to show you basically an example of how we incorporate some of those enhanced sampling techniques in MSLD so that we can extend the applicability of MSLD to traditionally challenging cases. So the system here I studied is just a protein. It's just a small molecule buried inside the protein. The systems are probably not irrelevant here, but I want to point out that this molecule contains an ionizable group. It's sort of easy to predict its protonation state in the solvent, but it's very hard to predict its protonation state when it's buried inside the protein core. And we want to use free energy calculations to basically estimate and to say, okay, what should be protonated or not. Okay. So if we do free energy calculations, what we found that, for example, if we run simulation once, this molecule is sort of trapped in this protonated. And if we run it again, it's trapped in the unprotonated state. So it's trapped in either of those end states. We couldn't sample those. We couldn't sample both end states reversibly. So that's why it's very hard to use this estimator here to compute the free energy, or almost impossible in this case because we didn't sample the other states at all. Okay, so that's the challenge we have here. And then if we we want to ask basically, we want to ask basically what's the fundamental reason of this no free energy calculation. And what we found is that in one of my simulation, it's sort of trapped in protonated state, so that's why it acts as a hydrogen bound donor. and it forms very strong interactions with this oxygen here in this asparagine group. And in another case, when this molecule is trapped in the unprotonated state, it forms a different hydrogen bond with this asparagine 177. So in other words, what we are observing is that once we have buried the charges inside the protein, it may form very strong, for example, hydrogen bonding interaction network. And if we look at the time scale here, it takes at least tens of nanoseconds to allow this structure reorganization to happen. So in other words, this slow conformation is the main reason why we see slow convergence in free energy calculus. So that's why we want to say, can we do something to accelerate this type of conformational changes? Yeah. And the idea we use something very simple. It's called replica exchange. I think most of you are probably very familiar with this, but I just want to remind you, we just run multiple simulations under different conditions and periodically exchange them. Two key ideas here is that first, under these perturbed conditions, we're able to see conformational changes more rapidly. And another thing is, since we use Metropolis criteria, we're able to recover the correct ensemble in the end. So inspired by this, still in this framework, what we design is here. In these perturbed conditions, what we do is we're going to scale down basically the partial charges of those residues that are important for this hydrogen bond interaction network. Because once the partial charges are reduced, then we think it will allow the breaking and reforming of hydrogen bonds more easily. So that's the key idea here. Of course, we also still have replica exchange and with Metropolis criteria to make sure in the end we still get the correct result. So we just named this method like a reject. A very funny name, I think. So how does it work? So what we found is that in original simulations, we are sort of trapped in alchemical space, for example, in unbound state, in the protonated state. But now with reject, we're able to see sampling of both end states, both protonated and unprotonated state. And also in terms of structures, we're able to see, of course, more structural reorganization here. So that's good. And we also want to see, can we extend this to basically a more challenging system? So here, the system that I studied is I have a buried salt bridge within the protein. So this is commonly found, for example, in ion channels or biocatalysts and a lot of other systems. So it's sort of like relevant to a lot of other problems there. And we're interested in is, what the question is, will this salt bridge stay basically charged or neutral inside this protein core? Okay. What we found is that, of course, like not very surprisingly, in original MSLD simulations, it's very hard to sample both end states. For example, for this glutamate residue, it's sort of trapped in this proteinated state. And for this lysine, we are not able to sample both end states a lot. But with reject, we are able to see a lot of sampling of both end states, which allow us to basically estimate free energies much rapidly. So I guess that's almost concludes my presentation today. I guess hopefully I have convinced you that we have built a computational framework, which is MSLD here. It's a very it's a physics-based high-throughput free energy method that can be used to to guide the need optimization process. And also by incorporating more enhanced sampling technique to accelerate those conformational changes, we are able to increase the efficiency of MSLD so that it can be applied to study traditionally challenging cases. So with that, I would like to thank my advisors, especially Dr. Brooks and all the other group members and my collaborator, Dr. Matthew Soner from their lab did the experimental work for my first project, TSS K1B project, and also another one, Dr. Jian Hanchen, who is my PhD advisor. So thank you very much, and I'm happy to take any questions.
Thank you so much. It was really exciting. Any questions? Hopefully we can get a couple questions from trainees.
I think there is one in the chat from Cheng Chin to maybe get things started, or maybe he'd like to unmute and ask it.
Yeah, so you use replica exchange simulation with metropolis criteria. So I suppose that's a Monte Carlo simulation. If yes, how was your movement defined?
Okay. That's a great question. Let me see. I want to go back to here. I want to go back to my illustration. So what we do is here we have first we run some molecular dynamic simulations for example for 2 picoseconds or something or some amount of time. Okay, we run multiple independent simulations there. Okay, so what we are going to do next is, for example, between replica 0 and replica 1. For replica 0 condition, I'm going to say should I change my condition to replica 1 condition? So I probably want to use an example. So in condition 0, for example, it can be 300K. This condition 1 can be 400K. So basically, I'm having increasing temperature here. Okay. So what I'm saying is, at the end of the simulation, should I change my temperature from 300K to 400K? And for replica 1, should I change correspondingly, should I change the temperature from, let's say, 400K back to 300K? So that's the part. Yeah.
So I think I probably didn't ask the question correctly. So this is replica exchange Monte Carlo or replica exchange molecular dynamics?
Oh, this is molecular dynamics. The Monte Carlo part is just a basic exchange of the conditions there. That's the metropolis criteria we use. Yeah, it's not the simulation itself is not Monte Carlo, it's the MD simulation. Yeah. Sorry for the confusion.
Okay. So the metropolis is just for the replica exchange part. Not
it's for the
within, right, not within each replica. Okay, that makes sense.
Yes, because here we run explicit solvent simulations. It's very hard to do Monte Carlo Monte Carlo simulation. Yeah. So okay. Yeah. Okay, thank you very much for your question.
More questions?
I have a question. For the replica exchange, can you go from like condition I to like any other condition, or does it have to be condition I to I+1?
In principle, yes, it can be any other condition. It's just in practice, usually what we do is only we exchange between neighboring conditions because the exchange ratio, the acceptance ratio will be higher there. If we do condition, as long as we didn't introduce any bias in our exchange, it can be anything. But if we do exchange between 0 and 3, very likely when it's going to be rejected, this movement is going to be rejected. This exchange is going to be rejected. So usually we just do neighboring exchange. Yeah.
So when you're doing the lambda being a free parameter rather than, say, like steps, if you have very different energy in one or the other, would you end up sampling in just, say, one of the molecules rather than the other? And is that going to be challenging to actually estimate the perturbation-free energy?
Yes, that's a great question. Actually, I didn't say that. I didn't say anything about that in my presentation. Actually, in this in the Hamiltonian of our system, we have an extra term there. It's called biasing potential free energy, which is a function of those lambdas. And the purpose of the extra term biasing potential energy there is to make sure there are several things. First, to bring those free energies of, for example, if they are completely different, they are so different. Well, we want to bring them to similar levels so that it is easier to sample them. Another thing is we want to reduce the barrier of those transitions so that we can make the sampling much easier. So for example, for some of those cases that I showed earlier here, so those bias impotential free energies are actually perfect. And even in that case, I still couldn't see sampling of both end states. So that's where we found like even like a perfect bias potential free energy is not sufficient. In those challenging cases, we need to basically accelerate conformational transitions in order to see those basically sampling, in order to get basically sufficient sampling. Yeah.
So I'm less familiar with these types of enhanced sampling approaches, but the one sort of small thing I do know is that sometimes there's a trade-off in terms of being able to better estimate the relative free energies versus estimating the kinetic barriers. And I'm curious if say, like, why would you choose this strategy over, say, some of the techniques that Tara's presented, or if other enhanced sampling techniques, and sort of what what is it about the system that you need to know or want to know in order to choose this type of method?
Thank you. That's a very insightful question. And actually, I was at Tara's seminar today. She presented a beautiful work. She used this weighted ensemble method. She can get both kinetics and basically thermodynamics quantities out there. Okay, but here in my case, we actually don't have we couldn't estimate the kinetic parameters there. So all the quantities we care in this case are just thermodynamic quantities, basically the free energy there.
Right, because the thermodynamics the kinetics are not real, right? There's no molecule that actually
it's all the kinetics here are messed up. We already accelerate all of those transitions, so kinetics are already messed up. So that's why we already gave up in some sense. Like, but the thing is, we want to. So in this case, the reason why I sort of like this approach is like we can do only minimum changes, basically very simple changes of the system by basically simply reducing partial charges of few residues, so that we can accelerate a lot of those transitions. So I think in some sense, it's a very simple but a very effective approach. I mean, we can do others. We can do more complicated cases, but I think this is already like a simple enough but efficient enough at the same time. So that's the thing here. I mean, but I think it depends on the problem we are interested in, because here we are just interested in like, is this proteinated state more favorable than the, for example, the other unproteinated state? So that's the thing we care. So that's why we can make a lot of assumptions and there.
So one thing that people use the FEP when they do the explicit transitions, they often build these networks of different transitions, and then you can there's some self-consistency conditions where you say if you go all the way around a cycle, it should be 0, and so if it's not, then you know that something is off. I'm curious, when you do all these different ones in the same run, if you can also kind of extract the self-consistency criteria, and if that gives you a way of sort of checking the validity of the simulations.
Thank you. That's a great question. Yes, in FEP, usually they do have this, for example, cycle closure thing to check basically how the uncertainty or the level of precision of their result. But here if we also think about this transition maps, in principle, we allowed all possible transitions between any two molecules. So that's the thing. Okay, so that's why, like in this case, we don't really do cycle closure type of check to check the uncertainty. But what we usually do is like for example, we run multiple simulations. We run multiple independent runs, or for example, we start from different structures. Okay, in the end, will my result will my result be dependent on my starting structure, or will my multiple independent runs will give similar results within the uncertainty? So those are usually the type of checks we do to check the convergence or check the answer or get some estimation of uncertainty there. So it's like a yeah, I think it's like it's just the previous cycle closure thing doesn't apply here, I think. Yeah. So usually, for example, here we usually at least run 5 independent runs, and then do bootstrapping and then see what's the uncertainty. We get the average, we get the uncertainty over there. So that's what we do. Yeah.
Nice. Any final questions? Okay, well, let's thank Xiaorong for a really detailed presentation. It's really exciting to see one of the largest free energy perturbation experiments ever run. So thank you for sharing.
Thank you. Thank you. Let me stop sharing.
Welcome, everybody.
Thank you.
We're a few people that came in. I saw Peter, you're there. Welcome, Lydia. So one of the things we were thinking about is that we've built this community around these meetings, and I've been having a lot of fun with them, and I hope you have too. But we were thinking about maybe other things that we might want to do beyond just having monthly meetings. And one of the ideas that we floated was maybe doing some sort of like one day or maybe even a weekend hackathon where we get together and as groups and try to solve problems. So learn new methods or apply it to new things. And I think there's a lot of really exciting things we could do out there, but we were hoping that maybe we could brainstorm a little bit about what people were looking for in terms of the community, what type of applications, new methods, what they would like to see out of a hackathon, and that would sort of shape the parameters in terms of what types of projects we could go after. Yeah, go ahead, Lydia. I know you had a lot.
Well, I was hoping, I like the hackathon idea, but I was hoping maybe we could open the question even more broadly and just talk about formats. What formats would people find useful just in general? What do you want to be talking about at these meetings? Because I think for me, at least, it's been great to see the kinds of presentations that we've been seeing, and I think we should keep that going to some extent, but I wonder if we should also alternate formats. Maybe even having like a mini kind of just coding session. You can call it a mini hackathon if you want to, but just at some of the weeks of our meetings also would be good, but maybe people just like this the way it is. I don't know. I guess I would love to hear what people would find useful, because that's really what this is all about, right? It's not about just like us making people show up for something, but what do people want to see?
I really like the presentations we've had so far, but could we maybe do a mini journal club kind of thing once in a while, maybe reading papers?
Yeah, that's a really good idea. I like that a lot. Ching-Ching, I see you on meeting as well.
Yeah, so especially for new methodologies in the field that people in this community may want to use, I think it's also good to have some journal presentation of 15 or half an hour long.
Well, one thing we could do, I guess, is we could make it so that we had like either 1 week journal club, 1 week talks, or we could have and that's probably better, because my other idea would be like split the time half and half, but that's probably not enough time for a reasonable journal club with discussions. So maybe we should just work those into our cycle of having a journal club week a journal club month once every couple months.
So how long do you feel a journal club should be? So, because I really thought that like half an hour like other talks is sufficient.
The way
Well,
that
I
we
think-
journal
go ahead.
club is more interactive, where we let people talk about different parts of the figure rather than just somebody presenting paper as a talk. I think that interactivity is really good, and it helps be more engaging, although it does take longer to get through. So I would prefer to have a full hour to do a paper and really talk about like possible experimental choices and really get into it, rather than trying to just race through and hit the highlights. I feel like that's what you can get from reading a paper, but the journal clubs are more interesting if people can have a discussion around it.
Yeah, and I think that's especially true with a group like this, where we have such a broad range of expertise and interests represented. that we can really talk about both the serious technicalities and the applications, and yes, experimental connections. So that's something we can easily work into the series, I would think. It's a good idea. Thank you.
One of the other formats that we were thinking of that would be fun is to have more chalk talk style. So people were thinking about proposals or, wanting to sort of bounce ideas off of around doing something. Peter, I know you've run chalk talks for, MDD. Do you have any suggestions on sort of what's the most useful, or, what would people like to see in terms of kind of more interactive sort of discussion rather than more extemporaneous talks, rather than, sort of polished, really high quality stuff that like we've been seeing so far?
Right. Yeah. I mean, these are all excellent suggestions, and I'm probably the last person who should influence the direction of this group, since, to be perfectly frank, most of what you talk about is way over my head. But I mean, so that sort of begs the question, why am I here? And so I think, certainly to learn, right? And a lot of that is learning, not necessarily the field, but what are my colleagues doing here at Michigan? What are the exciting areas of research that are ongoing that are sort of adjacent to what I'm actually doing? And related to that, what are the opportunities for potential collaboration where perhaps my skills are complementary to somebody else's in the group? And so I think with the presentation styles, they've been great. I've certainly I have learned, and perhaps not as much as others because I'm starting from a lower base, but they've been really interesting talks, the ones I've been able to attend. But I don't think we and maybe others have, but I don't think there's been so much discussion of how that work might lead to collaborations or joint projects amongst people in the group, or even outside the group, across the campus. So I guess that would be just something to think about. And certainly the more opportunities there are for people to get together and interact, the more likely that is to occur.
Well, so Matt, can I just clarify what you mean by chalk talk? Because I think trainees may not think about them in the same way that you and I think about them, where chalk talk is not just I show up with a whiteboard marker and talk about my research instead of showing up with slides, but rather it's supposed to be a forward-thinking kind of thing where you show what you're planning to do, and you really sketch out conceptually what you're planning to do in some particular area, and then you could get some constructive input on it.
Yeah,
I like the idea for the format. I just wanted to clarify that that's what we're talking about. It's not just banning slides. It's about being forward thinking. You can have a chalk talk actually with slides. That's how all my job talks were. But it's more about what you're presenting and how you're presenting it.
Absolutely. Yeah, it's sort of like you've got aims for a proposal, and you say, here's what I want to do here, this is what I'm going to do there. And then you get some like, well, that's not going to work, or this is, dependent, or, you could bring in these types of data or these applications. I think it can be really stimulating to get that type of feedback. It's, it's at least for faculty candidates that come, it's really exciting to see what they are doing. And it's unfortunate that, we sort of have to become invited to these things, but I should be more broadly available to practice and get these types of experiences. So I think it would be fun to sort of make it more accessible, and definitely there's so much different experiences and approaches that I think there could be really good feedback in a lot of different ways.
And I think we should give a prize to whoever can come up with an alternate to the phrase chalk talk, because we haven't used chalk in probably 30 years. So I'm gonna go out and go on a limb here and invite Tara to give her suggestions or thoughts, because I can see she's there and she's very quiet. So
Hi, sorry, I'm very distracted by the snow. I'm just looking out the window. Yeah, I agree with what everyone has said. I think finding a way to have a more interactive format could be nice. Some sort of even maybe tutorials where people can share methods with each other. And yeah, I think having the idea of fostering collaborations and starting to think of just like ideas where there might be room for for collaborative projects, whether or not they become like really large projects or smaller ones. Yeah.
What if there was some tutorials, what are things that people would want to learn about?
I think one kind of tutorial are just programs that we personally develop. So, especially for trainees nowadays, we need to publish papers in NAR or bioinformatics that requires a web server plus a software package. And many beginners are not familiar with how to show their result and how to test them and allow other people to test them. So if before their submission, they can show other people their works around, that it may be a good way for other people to give them feedback and to find out different bugs.
You think that maybe what would make sense would be for us to just pilot a couple of these different format ideas, maybe for those of us who are more on the organization side, once we get through our currently planned slate of talks, which I think runs for a couple more months, maybe we could have someone volunteer as tribute to give a chalk talk, and someone lead a journal club, and someone lead kind of a hackathon, and someone lead a tutorial the way Cheng-Shin was talking about. And I'm pretty sure I know who's getting volunteered for that one. And we could just see how they go and then revisit the idea or ideas.
Yeah, I think that sounds like a lot of fun.
Okay, so maybe Matt, if it's okay with you, I might just start adding some stuff to the schedule where we would allocate a month for each of these types of sessions and either find a victim or volunteer myself, and we could try some of them out.
For sure. Yeah, if people want to volunteer or have ideas, there's lots of room for leadership opportunities. So we're barely holding it together, but I'm really excited to see you all each month, and we're thrilled that this is survived as long as it has. So I would like to see it grow and more things happen, which would be great. So if you're excited to get involved and want to reach out, and we'll it's a spreadsheet. I don't know, admin access is the right word. It's like, so, but please, we would love to be in, have more people involved in getting this organized. Okay, well, get home safe.
Okay.
Looks like a lot of snow out there.
You
So
want to say something else, Peter? Did you have?
No, just saying, sounds good. I'm offering encouragement and support.
And it's appreciated. Thank you.
Yeah, thank you both for leading this and making it keep going. I really like it.
fantastic.
I'm really glad to hear that. Everyone be super careful on the roads if you have to travel anywhere, because it is a mess out there.
So the other thing we could potentially think of is, if we wanted to do like a retreat or something offsite, that might take funds, but there is pots of money around. So if somebody's interested in seeing that happen and wants to think about, shopping it around to different, get sponsorship from MICDE or Midas or MDD or something, we can see if there's things to apply for to get enough money to bring everybody to,
Would that be like
Maui Bay.
visiting another institution, or like
It
what's
might be
the?
like going to Maui Bay and doing like an offsite retreat with this. Might be fun.
Oh.
But anyway, it's money. So, but
Yeah.
yeah, you think about what we could do. Okay, just planting the seed. It's great to see everybody. Thank you for coming. Thank you again, Xiaorong, for presenting, and we'll see you next time.
Thank you. Bye.
Bye.